All right. Thank you for joining us today, right before our sort of first long weekend of the summer. And welcome, everyone. It's a real pleasure today to introduce Ahilyah Sawh to present her work on chromosome acrobatics during development. Ahilyah did her Ph.D. in biochemistry at McGill University and then postdoctoral work at Harvard University and followed the lab to Basel. And then started in 2003 at the University of Toronto as an assistant professor in the Department of Biochemistry, where she also holds a Canada Research Chair in 4D genomics. So thank you for joining us today, Ahilyah, and I will turn it over to you.
Great. Thank you, Jennifer. I'm just going to share my screen. Hopefully it looks good. How's that? Is that okay? Okay, good.
It looks good.
Okay, thanks. So I'm super happy to present to you today, tag teaming with Nour. So first time I've done a joint seminar with one of my students. Nour is my first PhD student, so it's really exciting. So what I will do in my portion of the seminar is give you like some background information and a bit of a historical overview that led us to where we are today and the questions that we're pursuing in my lab. So as Jennifer said, we're just 2 years into my tenure as an assistant professor, and Nour is a little bit over a year into her tenure as a PhD student. So it's early days for both of us, but still like super exciting. So I'll get started. I'm just gonna move some windows around. There. Okay. So, as Jennifer said, I'm a biochemist, and a biochemist's favorite question is how does structure relate to function? And so that's what we are also interested in as well, but instead of looking at a particular protein or particular family of proteins, we're interested in the entire organelle of the nucleus, which I think is one of the most fascinating or the most fascinating organelles of them all because it's kind of a black box. And specifically, the questions that we're interested in are how the genome is physically organized and how this organization influences its function. And so this organization is actually a monumental logistical and mechanical challenge that every one of our nuclei have to deal with, which is compacting meters and meters of DNA into the nucleus, which is only microns in diameter, right? And this compaction cannot be completely random or knotted or misfolded because we know that the nucleus has to maintain essential functions, including transcription, replication, recombination, and repair. And so when we like even simplify it even more, I like to ask students, or not even students, everyone, I like to ask them the question, what do chromosomes look like? And usually the response I get after a bit of stammering is some sort of X-shaped thing that you would see in your textbooks or on Google. If you Google it, you will see all these X shapes. But is this really true? And when is this true, right? This is only true during cell division. But we are interested in what's happening in interphase, actually. So an interphase is when cool stuff like transcription and replication and repair happen. And if you look in your textbooks, what you'll see is a bit of a fanciful drawing or cartoons of a bowl of spaghetti, for example, of what chromatin looks like, but this is not actually true. And so we want to really figure out what's going on here, what do chromosomes look like, and how does this relate to their function? And so what do we actually know about like what chromatin looks like in interface of the cell? So of course we know that DNA wraps around histones to form nucleosomes, and we've known this since the 1970s. But only in the past few decades did we gain any real like sequence-specific knowledge about what type of structures organize chromatin on larger and larger scales. And those include loops that can occur between regulatory elements like enhancers and promoters, And then on a larger scale, we have these elements called topologically associating domains, which come in typically two flavors, inactive or active. And on an even larger scale, we have different TADs coming together in space over large 3D distances in the nucleus to form these multi-megabase compartments. And we call them either active A compartment or inactive B compartment. And then finally, we refer to the volume that the whole chromosome takes up in the nucleus as its territory. And chromosomes can interact with other structures in the nucleus, like other chromosomes or the nuclear lamina along the edges of these territories. And so these, as well as other structures, organize chromatin on larger and larger scales, and they're increasingly being implicated in the function of the chromatin. Especially at the lower end. So most of the field is studying nuclear organization at the local scale of accessibility of chromatin, of loops and TADs. And then we're starting to learn a lot more about the mechanisms that generate these types of conformations and the functions that they might have in cells as well as in animals. On the other hand, we have comparatively very little information about the larger scale of things. So what's happening at the level of compartments and territories and whole genome. And so these are more black box than the lower end of the scale, and what I'm more interested in as when I started my postdoc and started working on nuclear organization, it's the larger end of things that I really wanted to focus on. And in particular, we have even less information in vivo. So most of the field studies chromosome organization in cultured cells or in particular cell lines where they differentiate. But what about in animals? What do we know about how chromosomes look like and what they do in animals? It's a lot less, actually. So the first evidence actually comes from the cytological level. If you look at EM images of nuclei, you'll see two different classes of chromatin based on how the DNA or chromatin stains. So we have this darkly stained euchromatin, or sorry, darkly stained heterochromatin, which corresponds to the B compartment, so the inactive stuff. And we have lightly stained euchromatin, which corresponds to the A compartment or active stuff. And it's thought that the B compartment chromatin has a higher affinity for the nuclear lamina, which is thought also to be a repressive environment. Now, in terms of the functions of these compartments, canonically it's thought that these two compartments are involved in gene regulation or transcriptional regulation. And that's because they're enriched for these different sequences that are either active or inactive. However, when we look in more and more different biological contexts and in different types of tissues, we start to discover new and unexpected functions for these compartments. And my favorite example of this, the surprising function, it comes from the rod photoreceptors of nocturnal animals like mice and other rodents. And so in most cells that have been looked at so far, what we've seen is this similar organization to what I showed you in this EM image right here, where you have B compartment chromatin making up the periphery of the nucleus and interacting with the lamina, and A compartment chromatin making up the center. We also have some B compartment chromatin surrounding nucleoli. But this is the conventional way of thinking of how A and B are organized radially. However, in the rod photoreceptors of these mice, what happens is this dramatic inversion of the radial organization, where now we have this super compact center of B compartment chromatin, and then the A is relegated to the periphery of the nucleus. Now, based on the canonical roles of what we think euchromatin and heterochromatin would do, people have predicted that this would change the transcriptional profiles of the cells completely, but that's not what happened. There were very few transcriptional changes, and in fact, the real function for this inversion was a more biophysical one. So this inversion transformed these photoreceptors into, or the nuclei of these photoreceptors into micro lenses that help to propagate light and to facilitate nocturnal vision in the animal. And this was like wild and crazy, but also super cool. And it kind of highlights the fact that we've just scratched the surface in terms of what we know about what compartments actually look like in different biological contexts and what the physiological functions of them are. Now, my favorite biological context is actually embryogenesis, because I think it's a super exciting time to be alive for an organism, because you're going from a very pluripotent state to something super complicated and differentiated of different tissues that interact with one another and cooperate to make an organism. And so when we look during embryogenesis in C. elegans, which is our favorite model organism, we can see that if you go from the early stages all the way to a more differentiated state, you have this gradual segregation of euchromatin and heterochromatin, marked by these dark regions for heterochromatin and light regions for euchromatin. And this gradual separation or segregation corresponds to the gradual loss of plasticity that happens in the embryo and the adoption of new sulfates and different sulfates in different cells. And so what this type of information gives us is really good temporal and spatial understanding into the segregation of these two types of chromatin. But of course, what we're missing is any sequence specificity. So we don't know where our favorite genes are, and we don't know what class they're in any of these cases in these cytological experiments. And on the other hand, what we have is evidence that comes from the biochemical side. So I don't need to explain Hi-C to this audience, I think, but based on mainly chromosome conformation capture techniques, ranging from 3C to Hi-C and 5C and 4C and all those, we have actually sequence-specific information about what type of contacts exist in cells and in embryos. So in C. elegans, this was done, and these labs, among others, were able to determine the different TADs, so the topologically associating domains and compartments in C. elegans chromosomes. And so what you need to know is that in C. elegans, for example, we have this a very stereotyped 2D arrangement of compartments on autosomes. And this is an example with chromosome 5, for example. So in C. elegans, the centers of the autosomes make up a large A compartment, and they are flanked by 2 B compartments in the arms. And so those correspond to euchromatin and heterochromatin enriched regions on the chromosome. Now this type of information really gave us exquisite sequence specificity, but of course we're lacking any spatial or temporal context into where these things are in different cells and how it changes over time, because most of this work has been done in bulk embryos, for example. And so this is where I came into the picture when I started my postdoc, because what I wanted to do was really like bridge the gap between these two types of approaches and try to understand with sequence specificity, what do chromosomes look like? And then from there, can we figure out what they do? And so specifically what I wanted to do was to trace the 3D trajectories of chromosomes in situ. And this would allow me to combine the spatial and temporal context with the sequence specificity that we had from sequencing, as well as the cytological methods. And I was also super interested in understanding how conformation varies at the single molecule level, from cell to cell, from embryo to embryo. Even within the same nucleus, how does conformation vary? And this is lacking in the field because most studies had been conducted on bulk samples, as I mentioned before. So how did we decide to do this? We decided to use image-based spatial omics to directly measure chromosome structure in embryos. And the specific technique we used was chromosome tracing by multiplex sequential FISH. And this is a technique that was invented by my friend and collaborator, Stephen Wang, when he was a postdoc in the Zhuang lab at Harvard. And he invented this to work in cultured cells, and then I joined him and took it into embryos in an animal model. And so how this works is we do highly multiplexed fish on our samples, be it whatever you want. We wanted to do it on C. elegans embryos, but you can do it on your favorite sample as well. So how it works is if you imagine that this is your chromosome in 2D, and then this is your chromosome in 3D, it forms these TADs, which are some are bigger, some are smaller, some are active, some are inactive. And we want to map all of these things, right? So the first step is we label and image the entire chromosome. And so we accomplish this by using a large pool of labeled oligonucleotides as our FISH probes, and these hybridize to the entire length of the chromosome. So when you image with these probes, what you see is the entire chromosome territory. And this is great, so we can get an idea of what the chromosome looks like globally, but we don't actually have like information about like your favorite region or your favorite gene, right? So what we do next is we label an image with individual probes that are specific to different regions along the length of the chromosome. So what I want you to imagine is that you shrunk yourself down into a tiny nucleus and you're walking along the length of the chromosome and dropping breadcrumbs as you go to remember the path that you've taken. And so with this way, we can both have the global organization of the chromosome territory itself, and within that territory, we know exactly where different regions of that chromosome are with sequence specificity and high spatial resolution. And so the experiment is fully automated because nobody wants to sit on the microscope for days or weeks at a time. And it happens while the sample is sitting on the microscope here and hooked up to a microfluidic system composed of multiple valve positioners and a peristaltic pump. And then we control everything with the computer, and we tell the microscope when to image and then when to wash away the probes from a particular round and follow it up with the next round of imaging and or hybridization and imaging for the next sequences that you're interested in. And so this is what the data actually looks like for a pretty simple example. So what you're looking at is a C. elegans embryo with about 30 cells. So that's the DAPI is telling you the nuclei. And then this image here is of the chromosome territories. So that's with the primary probe that I mentioned before. So what you're seeing are blobs. And the blobs represent the territories of all the chromosome 1 chromosome 1 copies in this embryo. C. elegans are diploid and dividing and rapidly growing. So you'll see between 2 and 4 blobs for each chromosome in each nucleus. And in this experiment, what I did is I had labeled 14 regions along the length of chromosome 1. And then we did the sequential FISH on those regions, and this is the type of data that you'll get. So for each region, you'll see a number of dots, and each dot corresponds to that particular region within chromosome 1, within each copy of chromosome 1 in the whole embryo. And so what we do next is then we combine all the data together and overlay all those dots on top of one another. And then we can directly reconstruct the trajectories of each of those chromosomes inside the cells. And so what you can see here is that we can go from the single molecule scale all the way to the whole organismal scale in the same experiment, which is super powerful. And what I didn't mention is that we use actually Gaussian fitting to each one of these dots that you see, which allows us to get subpixel super resolution in terms of spatial resolution. And so we can really look at things that are super close together. Here in this example, we have regions that are about a megabase apart or 0.5 to 1 megabase apart. But we can go down to things that are 2 kilobases apart and tell them and distinguish them from one another. So it's a super powerful technique that we can use to answer different questions that we're interested in. And so what are the types of things that we can learn from these experiments or this type of approach? We don't want to just get pretty pictures and look at them. We want to find out things about chromosomes, right? So The first questions that we were interested in were what do intrachromosomal conformations look like in embryos? But we're not limited to only looking at inside a particular chromosome. You can label as many things as you want inside your cells, and you can also interrogate interchromosomal interactions. Because chromosome tracing by multiplex FISH is a in situ technique, we preserve all the cellular and organismal features of the sample. That means we can measure them. So for example, we preserve the lamina interaction of the chromosome so we can see what relationship the lamina has to our different conformations. And then finally, since it's a high throughput in situ or single molecule technique, what we can do is measure chromosome to chromosome variability with a lot of power and much more power than we had before by doing single FISH experiments on a couple of different regions or by doing Hi-C in bulk. And so this was really like a game changer to understand the variability that can happen in vivo. And so what I wanted to talk to you a little bit about is some of the work that I did during my postdoc that gave us some surprising results and then led us to the questions that Nour is going to be answering in her PhD. So I'll talk to you a little bit about chromosome 5, which was my favorite chromosome when I started working on this project. And that's only because it's the biggest one. And we didn't know if we would actually find anything interesting. So we decided to look at the biggest chromosome in C. elegans and see if we can measure any distances and determine what type of conformations were there. So when we do multiplex FISH, we label all these different regions along the length of the chromosome. And then we actually measure the distances, the pairwise distances between all possible pairs. So not just 1 and 2, but 1 and 3, 5, 9, 20, whatever, and then all possible combinations. And we produce a distance matrix that looks like this. So here what you're looking at is regions 1 to 22 versus regions 1 to 22. And it's displayed as a color heat map, very similar to Hi-C data, where things that are closer together are darker and things that are further apart are lighter. So what we can learn from this is which regions. Here, the regions are particularly looking at individual TADs. So there are 22 TADs in chromosome 5, and we're looking at TAD-to-TAD interactions in this experiment. So that means we're measuring compartmentalization. So which TADs come together into which compartments. And so when we did experiments for chromosome 5 in early embryos, we found this sort of result. And what we saw is that the two arms of the chromosome were more compact than the center for sure. And this corresponds to the previous assignment of these regions as A or B compartments. But what we didn't expect was that the two arms were far apart in space. And so this means that the two B compartments were not consolidated, they were not together, they were actually completely polarized in space. And we confirmed this with higher resolution tracing to confirm that these regions for sure in the arms are more compact than the centers, which meant that in the early embryo, we had this unconventional organization in space of compartments where two B compartments are separated by a large active center. Now this was like interesting, but it's the bulk is the total number of chromosome traces that came from about 1,000 different chromosome traces. And what I mentioned to you before is that we're interested in variability, right? So from trace to trace, what are the differences that we could see? And how does conformation vary between individual chromosomes, and are compartments actually static? So are the same regions in chromosome 5 always compact, or do they open up at some point in time? And what could that mean? And so to answer this question, what we did is we treated our chromosome traces like single-cell RNA-seq data, basically, and we clustered them in PC space based on their TAD-to-TAD distances. So for chromosome 5, for example, we measured 22 TAD spots, basically, and all combinations of TAD spots would give us 220 variables to work with. And when we cluster those traces based on their tad-to-tad distances for chromosome 5 data, what we came out with was 4 subpopulations. And these subpopulations or clusters are basically clusters of chromosomes that are defined by their shape. And so now the question is, of course, what do they look like, right? So how do they compare to each other, and how do they compare to the population average? So remember that this is the population average. It's a barbell-like structure where the two B compartment arms are more compact and separated in space. Now, how does that compare to the single molecule data where we clustered them based on their shape? So the first cluster, which we called alpha, had a super compact left half of the chromosome and a more open right half of the chromosome. Beta was almost the mirror image of alpha, with a super compact right half of the chromosome and an open left half. Gamma had similar compaction between its two arms, so here and here. And it was also folded over in space, and now we have the two arms actually close together in space and interacting. And this was actually the structure that we would have predicted based on the previous Hi-C data, where we have these two arm regions classified as B compartments and close together in space. But now we know from the single molecule data that this type of conformation only occurs in 24% of the cases in early embryos. And then finally, cluster delta, the fourth one, was the strangest one of all and had very wide open loopy structure. And so what we concluded from this data was that compartmentalization is definitely not static from cell to cell, from embryo to embryo, and that compartmentalization is much more fluid than we thought it would be in the first place. And so chromosomes definitely adopt diverse compartment structures in early embryos, and all 4 of these different classes can coexist in the same stages. So even from the 4-cell stage all the way to the 40-cell stage, all were present at the same time, but maybe in different proportions. And so in addition to identifying these different conformations and looking what they look like, we can delve deeper into the single molecule data and identify which regions of those chromosomes are the most variable versus least variable. So what you're seeing here are the ones that are most variable are high on this dispersion plot. So these are the relationships between TADs, basically. So what you're seeing is that regions 2 and 4 are very variable. However, things that are close to regions 2 and 4 can have the opposite type of behavior. So regions 3 and 4 are actually the lowest variability amongst those TAD-to-TAD distance pairs. We can also look at individual clusters and define which regions are those marker regions. So marker regions meaning that they are significantly different within a particular cluster versus all other chromosomes in the population or other clusters themselves. And so what we can find is that for alpha, for example, the markers are in the left arm, opposite in beta. In gamma, we have markers in between the two arms themselves. So those are the ones that are coming together in space, and that actually defines that cluster as unique. And then in delta, the markers were all over the chromosome because they're significantly different in terms of distance versus all the rest. So they're very more wide and spread apart. And so this was actually super interesting because it allowed us to make quantitative comparisons of conformations in vivo at the single molecule level for really the first time. And we can use this in the future to try to understand which parts of the chromosome are really driving the different conformations. And so we have these different conformations, and one of the first questions we wanted to ask was, what different biological mechanisms could be generating them? So they all look quite different, and they look like they would do different things, but how were they made? And we thought that one really good candidate for defining one cluster versus another could be the interaction with the lamina, which I mentioned before. So we know that in all organisms, basically, B compartment chromatin is more enriched for lamina association, and this is what you see here. See here also for chromosome 5. The two arms of the chromosome by ChIP-seq interact more with the lamina. And so we know that these are the regions that are preferentially interacting with the lamina, but what we didn't know was how lamina interaction actually shapes whole chromosome conformation. So we decided to test this by using a mutant for a protein called Sec4. So Sec4 in C. elegans binds a heterochromatin marker, H3K9 trimethylation. And when you delete Sec4, what you do is you cut the cord basically between the chromosome and the lamina itself. So it's a very specific mutation we can make, which doesn't affect the level of the heterochromatin marker, or sorry, the histone modification, but it affects where it localizes in the cells. And so we're cutting the cord, so it's a physical disruption of where the chromatin is. And so then if we do chromosome tracing, we can find out what happens to the structure of the chromosome. And what we found is that actually the whole volume of the chromosome shrank. And when we look at the actual structures of the chromosomes, we can see that basically all the tad-to-tad distances were shrinking in size. And this resulted in the chromosome having a much more crumpled conformation, where the arms are now much closer together and the whole chromosome itself is shrunk. And so this led us to a model where we think that in wild-type situations, we think that lamina interaction actually systemically stretches the chromosome, which was unexpected for sure. And so what I'm showing you here is the population data, but what happens at the single chromosome level, as I mentioned before. So remember, in wild-type situation, we have these 4 different clusters or 4 different classes of chromosomes. And in the Sec4 mutants, when we did chromosome tracing and then clustered the data, we found several interesting things. So first of all, we lost cluster delta. Now, delta was the one that was the more wide open and stretched. And when we lose Sec4, we actually lost that cluster. So that's in line with our model that the actual interaction with the lamina is the thing that's stretching the chromosome to make larger conformations. Now, in terms of the other conformations, what we found in Sec4 mutants were 3 classes that were similar, but not exact, but very similar to alpha, beta, and gamma, but they were much more compact, also in line with the chromosome stretching idea. Another thing that we noticed is that for alpha and beta type chromosomes in particular, we lost this very sharp boundary that we had in wild-type situation between the compact regions of the chromosomes and the open regions. And now we have much more checkerboard type organization of looping, which means that the lamina interaction is actually is required for defining sharp boundaries of different compartments in the wild-type situation. And then finally, something really interesting we found was that the gamma type conformations in Sec4 mutants actually had the strongest B compartment compartmentalization scores compared to all other clusters in the other genotypes as well. And so you can see this here by this really strong interaction between the 2 arms of the chromosome compared to wild type, for example. And what this means is that actually the lamina interaction inhibits compartmentalization. So in wild-type situation, you have a less strong interaction between B compartment arms. And in the Sec4 situation, you have an enhanced interaction between those arms, leading to stronger B compartment interactions. And so we have all these different subtle effects of lamina and conformation. And I think these results really highlight or emphasize the advantages of looking at the single molecule level, because basically all of these effects were invisible if we had looked at the population level alone. And it highlights all the different nuances we could have in vivo because depending on what type of conformation you have a different interaction with the nuclear lamina and a different result if you are released from the lamina. And so what I want to do is just like summarize right now, and then Nour will take over. Um, we like to use our multiplex fish techniques to understand chromosome organization. I won't go too much into the other applications for multiplex fish, but just to mention that it's super flexible. We look at DNA, but we can look at RNA and protein as well. It's very flexible in terms of scale. So I showed you very simple examples of 14 regions or 22 regions along a couple different chromosomes. But Nour will describe how we're really massively scaling this up and going down in resolution as well to look at more smaller structures in addition to compartments. And we combine this with single molecule analysis to really understand the behavior of different chromosome molecules in vivo. And what I found during my postdoc were a lot of like really interesting things and follow-ups that we would, of course, love to follow up with in my own lab. And that includes developmental milestones. I didn't explain this to you, but we look at over development, what happens to chromosome organization. We found this new role for the lamina in systemically stretching chromosomes. And most interestingly to us is this high variability that we observed between different chromosomes of the same type, of the same age, same developmental age, but super different conformations and unique interactions with the lamina, for example. And so this leads us to a lot of new questions that we want to answer in the future, which I'll let Nour talk about. And basically, we want to know, what's the point? Why do we have these different conformations? What are they doing? And what does it mean for development? And so these are the questions that we'll pursue in my lab and using C. elegans and other models as our source material. And I'll let Nour take it over from there after I do my acknowledgements and answer a couple of questions. But we're super happy to be part of CIRC now back in Canada. It's really nice. I've had really great collaborators during my postdoc and continuing during the early years of my lab. We've been very grateful to have funding from these really nice institutions to get our lab started off on a really great foot. And importantly, the people in the lab are the best things. So Nour is one of many. And actually, we are in a mode of expansion. So we're hiring. So in case you're interested in this kind of work, we're looking for students, we're looking for postdocs. In particular, we're looking for people with a computational biology background. So if you're interested, just send me an email and I'll get back to you. And I'll be happy to take your questions.
Thank you, Ahilyah. That was fantastic. I just want to remind everybody to use the Q&A function for questions. And I see that there's already one in there from Sheila Teves, who is asking a question that I myself was also thinking about, which is, does the volume of nuclei change in the lamina mutants?
Very good question. So I was expecting it to maybe like go down, the volume of the nucleus, but actually they either don't change at all significantly or they're slightly bigger than in the wild type situation. So, and the slightly bigger happened in early embryogenesis. And this was a surprise because we thought that like, oh, that's strange. I thought like the nucleus would just shrink because the DNA is not taking up as much space as it was before. But we only looked at chromosome 5 in those experiments, so it's possible that there is some domino-like effects that's happening in other parts of the chromosomes or the genome, sorry, or other nuclear bodies are taking up this space and just filling it up even more, and that the interaction with the lamina was actually keeping nuclei smaller. So that's a pretty interesting question, and we don't know why. Yeah. So, but
Yeah, I agree that I was with you. I thought they would be smaller.
yeah.
We have another question in the Q&A, which says, hi, very beautiful spatial resolution during development. Do you see different transcriptional impacts of your variable alpha, beta, gamma, delta phenotypes? Does differential spatial organization impact function?
Yeah, that's the question for sure. So, during my postdoc, of course, we did not answer that question. We can speculate though. So actually, like based on polymer models and these sorts of things, which like take it with a grain of salt, alpha and beta would be what we would predict to be the more transcriptionally active based on the folding pattern of the chromosome itself. And but that's just a prediction. And so what Nour will talk about is how we're going to be testing whether one conformation is more transcriptionally active than another. And we do this in situ, and she will tell you.
Cool. Next question is, does gene expression change in the lamina mutants?
So for this particular mutant, it does change a little bit, but not a lot. So the SEC4 mutant is special because it doesn't change the histone modifications itself. So those are still there. So most things that are repressed stay repressed, and most things that are active stay active. With a few changes on chromosome 5 actually. So that chromosome is the one that's most affected by the loss of SEC4 in embryos. However, later in the life of the animal, we have more dramatic changes resulting from the loss of lamina attachment. So that could mean that in other stages, it's more important for gene expression regulation, but it is already established in the embryo itself. And I would also say that we don't know at the single cell level how gene expression changes for SEC4, which I think is a important question because we see all this single molecule variability with how lamina interaction influences chromosome conformation and maybe vice versa. So we have now just bulk information about gene expression changes, but I would expect that there would be a lot of variability from cell to cell.
Yeah, thank you for that. I think in the interest of time, we will move on to Nor Bazzi, who is a PhD student at the University of Toronto in the Sawh lab. And she's going to tell us about the future of chromosome acrobatics during development. Nour, please take it away and share your presentation.
Hello. Could you let me know what you see?
It looks good.
Looks good? Okay. Just gonna start from the beginning. Sorry, I'm trying to figure this out. Sorry. Okay, got it. Okay. Okay, hi everyone. My name is Nour. I'm a second year PhD student in Ahilyah's lab. So Ahilyah has been talking about exploring the molecular functions of conformations, and in our lab, we now want to take this a step further and explore function at the tissue level and at the organismal level and over time. So some of the burning questions we start to think about are what types of conformations are present, and is this different in different cell types? When and where do they appear throughout development? And how are they formed? And what do they do? Could some of them be more or less transcriptionally active than others? So I'm going to focus on the first two questions. And to answer these, we need to directly connect chromosome conformation, transcriptional output, and early cell fate decisions. And embryogenesis provides a natural set of dynamic conditions for us to study the general rules and principles that govern that relationship. And C. elegans is very well suited for the questions we want to ask because we can exploit its stereotyped and reproducible lineage. So the lineal relationship of any cell to any other cell is known, and the 3D positions of each cell is also stereotyped. So the two-cell stage here, the cell in green, yellow, or blue has the potential to become the germline, the intestine, muscle, pharynx, or a neuron. And each cell division follows a predictable pattern that results in a fixed number of cells with specific positions and specific fates. So each cell provides a natural set of conditions to ask and answer questions like, do cell type specific conformations exist? And are conformations inherited? Could they be reused? And so also, what is the sequence of events in different contexts? Could this be different in early versus later development? And to answer these questions, we need to be able to link chromosome conformation to cell type. And with co-immunofluorescence FISH, we can distinguish between germline chromosomes and somatic chromosomes. So these images follow an embryo throughout development from the zygote to the 16-cell stage. And here we're using IF to label PGL1, which is a germline-specific granule. And when we do this, and when we compare germline and somatic chromosome traces, we see that they are fundamentally different. So this t-SNE plot on the right shows that they separate quite well. And these distance matrices show inter and intra chromosomal interactions between chromosomes 1 and 2. In the germline cells, or the PGCs, chromosome 1 and 2 are more compact intrachromosomally and closer together interchromosomally when compared to the somatic cells. The germline cells are also consistently in contact with the lamina, whereas this is more variable in somatic cells. The germline cells have static conformations, so low variability, and the somatic cells have highly variable conformations. So what could be the driver of these differences? Well, we know that transcription is silent in the germline during early embryogenesis, and that the maternal to zygotic transition has not yet happened. So the maternal to zygotic transition is when developmental control is handed from the maternally provided gene products to those that are synthesized from the zygotic genome. And this happens in all animal embryos. And this transition is subdivided into two processes. So the red curve shows a degradation profile of maternal transcripts. And the light and dark blue curves show the minor and major waves of zygotic genome activation. So 2 slides ago, I pointed out the differences between the germline and the somatic conformations, and we know that transcription is silent in germline during early development. And now, in this new distance matrix on the right, I'm showing you the somatic signature before zygotic genome activation, so before transcription begins. And what's interesting here is that the somatic signature before ZGA is actually more similar to the germline signature than it is to the somatic signature after ZGA. So this indicates that the act of transcription during ZGA could be a major remodeler, and that perhaps the germline state is the default of a silent genome. So the next burning question is, could transcription alter chromosome conformation? And I'm going to do genetic perturbations using RNAi to test transcriptional impact. So first, to create a broad transcription inhibition across the entire embryo, I'm going to knock down AMA1, which is an essential subunit of Pol2. And then to induce transcription precociously in the germline, I want to knock down Pi1, which is a germline-specific protein that prevents transcription elongation. So next, I wanted to monitor the efficacy of these genetic perturbations. And so I stained for actively transcribing Pol2. And I'm showing you 3 channels here. So the DAPI for nuclear volume, P granules, which are a germline marker, and then active Pol2 immunostaining. So this is using a transcription elongation marker. And this is the empty vector negative control. The somatic cells are transcriptionally active. When we zoom in on the germline, we see that they are transcriptionally silent, and we know this to be true for the early embryo. Then we look at the AMA1 knockdown, and there is a loss of active Pol2 immunofluorescence in all cells. And then looking at the Pi1 knockdown, there is a gain in active Pol2 immunofluorescence in the germline. And so now with these conditions, we can begin to explore how transcription influences chromosome conformation. And here's some preliminary data. So this matrix shows the spatial distance changes in AMA1 RNAi minus the control empty vector. And the red regions are closer to each other in the AMA1 knockdown condition than in the empty vector. So these red regions are compact upon transcription inhibition. And we see evidence of that within and between chromosomes. But we were also surprised to find blue regions in chromosome 1 that decompact upon transcription inhibition. So it's becoming more and more apparent that there are genomic context-specific effects of transcription and that this is different in different chromosomes. So I'm going to continue to explore this relationship genome-wide over developmental time and, importantly, with cell type specificity. So one thing that's missing in our data so far is the cellular origin of each chromosome trace. So I've talked about using IF to identify the germline cell, but we want to be able to identify all cells and compare their genome organization. So I mentioned that the lineage is completely mapped, but identifying the cell IDs in each embryo is still a very daunting task. And in 1983, John Sulson did this manually by staring at embryos growing for hours and hours on end. We are not going to do that. It's not sustainable considering, we want to do this in a high throughput way. So we're talking hundreds to thousands of embryos. And previous methods for automated lineage mapping in C. elegans embryos require live imaging data where you need to track the movement of each nucleus. And this also involves a fair bit of manual curation and correction. But there are no methods to automatically do this from fixed image data where we take snapshots in time of embryos, and the prior history of each cell is not known. So I mentioned that the 3D positions of cells are stereotyped. And now we actually have a reference distance map. This was created by Ahilyah and her collaborators at the University of Basel. And it automates labeling by doing a 3D overlay of test images with ground truth images. So if we just label the germline precursor cell, so the one shown as P4 in this 16-cell embryo, then we can automatically identify all somatic cells. And then using these maps, so I should say, we can do this all the way from the 2-cell stage to the 360-cell stage. And then using these maps, now we can make more specific conclusions about conformation based on cell type. So I've shown you that loss of transcription increases germline signature, which is characterized by compact inter and intra-chromosomal interactions. And I've shown you that transcription causes some contradictory structural changes. And now we want to continue to explore that developmental dynamics of the conformation-transcription relationship by integrating conformation and single-cell RNA expression. And we also want to increase whole chromosome tracing resolution and scale by implementing MERFISH, which is multiplexed error-robust seq-fish. And this will allow us to look at, visualize smaller chromatin structures below the megabase pair scale, so things like tabs and loops, while maintaining our ability to look at compartments and territories. And we're going to integrate this method with transcription and look at all lineages over time for the ultimate goal of reaching a more comprehensive understanding of the relationship between the 4D genome, the transcriptome, and cell fate decisions. So thank you for listening. And I'd like to acknowledge my supervisor, Ahilyah, for guiding me through every step of the way. I also want to acknowledge Natalie in our lab for designing the fish probes. And thank you to my awesome committee members. And I also appreciate all the contributions made by all these great organizations who make this research possible. Thank you.
Thank you, Nour. That was really fantastic. It's going to be so exciting to see everything that you find from these systems. And if people have more questions, again, feel free to use the Q&A. There is one question, I think, that we sort of came in at the transition. So it says, have you investigated whether the removal of specific histone modifications leads to significant rearrangements in chromosome conformation?
So for me, was this one for Ahilyah, or should I?
I think either of you can jump in. if you have, either one of you can jump in and answer. Yeah.
Sure, sure. So we haven't done those experiments yet, but we're definitely interested in understanding, the building blocks of different conformations, and histone modifications are definitely like an obvious thing to look at for sure. But we haven't done it yet. But what I will tell you is that it has been done in C. elegans embryos, and then the readout being high C. And so. And the results were actually like a bit unexpected. So Barbara Meyer's lab has done high C with histone methyltransferase mutants that eliminate all H3K9 trimethylation and dimethylation, and found not very dramatic changes in compartmentalization. So the A and B compartments are still there. The boundaries are a little bit shifted compared to the wild type, but they're still there, which is unexpected and also surprising. So it could mean like multiple things, right? It could mean that that's not enough to disrupt compartmentalization, could mean that there is no direct relationship between those histone modifications themselves and compartmentalization. And that we have to cast a broader net, basically. So we're interested in general and what molecular factors drive different conformations. And so to look with a broader net, I think like some ideas are to do like a general screen for like anything nuclear and then find out what is the effect on compartmentalization. But definitely, I think it's likely that it's a combinatorial effect. So histone modification alone is not enough.
Thank you. We have a couple additional questions. So in mammalian cells, degrading Pol2 doesn't have a big impact on the 3D genome at steady state. Do you think Pol2 reshapes the 3D genome only during cell state transition?
Yeah, it's that's possible. Nour, you can go ahead and answer. Yeah, go ahead.
Okay. Sorry, I wasn't sure. Okay. Yeah, so when we do degrade Pol2, so when we do an AMO1 inhibition, and so AMO1 is an essential subunit of Pol2, we do see that the so the DAPI signal kind of becomes more like punctate and more compact, so kind of resembling the germline signature. So sorry, what do you mean by during at steady state? Sorry, could you clarify that?
So
I think, sorry.
yeah, it's okay.
I
So
was
what
just
was
going to say, I think
done?
they're wondering about the difference between like a cell in homeostasis versus
Oh,
a cell that's going through developmental transition.
oh, I see, I see. Yeah.
Yeah, it's possible that like the cells in early development are much more sensitive to these sorts of changes. One being Pol2 inhibition. I think what was done in previous work was high C also, based on I can't remember if they deleted Pol2 or inhibited with amanitin. So then you're also looking like on bulk, and if we look on bulk, we see in general compaction happening of the entire of chromosomes 1 and 2. That's what Nour showed you already, but also like some small changes in the opposite direction, right? So it's possible that at the bulk level, you dilute these changes that are apparent. What Nour didn't show you was that we can also cluster that data, and we have different conformations that go up and down depending on if transcription is on or off. So it's not like your whole genome changes completely when you lose transcription, but certain classes of conformations are enriched or not enriched in different situations. And what she's going to do in the future is try to connect that to which cells that's happening in. And then we can actually figure out, okay, but which transcripts are the ones that are the most affected and which are the most sensitive to conformation and transcriptional inhibition. So yeah, I think it's like context dependent is really the answer.
Yeah. I think just because we're just at time now, we have one quick sort of question that you may or may not have any information on, which is, do the spatial organizations alter further during aging of the organism?
We don't know.
Yeah.
Yeah, I think it has been looked at like so in aging you do have a breakdown in the nuclear lamina for sure. So I would predict that definitely things that are interacting with the lamina and dependent on the lamina interaction would be affected, and that would be interesting to look at for sure.
Yeah, for sure. Thank you. I think in the interest of time, we will call it there. And thank you both for joining us today and for presenting your really fantastic work. And I'm so looking forward to seeing more out of your team. It's going to be a really exciting few years
Thank
for sure.
you.
Thank you.
Thanks, Jennifer. Thanks everyone for attending. It was great.
Bye, everyone, and have a great long weekend.
Yeah.